All right, let's talk about something that drives every single assay developer crazy. A run failing because of high percent CV. You know what I'm talking about. It is a stubborn, frustrating problem that can stop your project dead in its tracks. But what if you could actually move past all that guesswork and start engineering real, robust solutions? Well, that is exactly what we are going to dive into today.
I mean, this is the big question, isn't it? It's the one that keeps you at the bench way too late. You did everything right, you followed the protocol, your standards look perfect, but your replicates are just all over the map. So what is it? Is it your pipetting, the reagents, the plate washer? The answer, as you've probably guessed, is usually a lot more complicated than you think.
And let's be really clear about what's at stake here. Getting a handle on your precision, which is what your CV is all about, isn't just some academic exercise. It is literally the difference between creating a validated clinical tool that could change lives and just pouring a ton of time and money into an expensive failure. Getting this right is everything.
Okay, so our investigation is going to start right here. We've got to learn how to read the clues that your data is already trying to show you. See those percent CV numbers? They're not just a pass or fail grade. They're actually a diagnostic signature, and they can tell you exactly what kind of problem you're dealing with.
The key to cracking that code is a concept with a pretty fancy name, heteroscedasticity. I know it sounds a little intimidating, but all it really means is that the noisiness or the variance in your assay isn't always the same. How that noise changes as your signal gets stronger, well, that's our biggest clue. So these two patterns are pretty much what you're going to see. Over on the left, you've got additive noise. This is when your CV is sky high at the low end of your curve, but then gets better and better as the signal goes up. That almost always points to something like background signal or nonspecific binding. But what if you have the graph on the right? A flat, frustrating, let's say 15% CV across the entire curve? That's a whole different animal. That's proportional noise. And this is crucial. That pattern almost always points to a physical issue, like inconsistent pipetting or a problem with your reagent concentrations. All right, now that we know how to classify the crime, let's round up the usual suspects. We can pretty much group all the sources of noise into two main categories. You've got your mechanical vectors, those are all about the physics of your process, and then you've got your biological vectors, which are all about the chemistry happening inside the well. You know, we all love to blame pipetting. It's the first thing we think of, but it's almost never just about having good hands. It's about physics. An air displacement pipette is super sensitive to temperature. As you work your way across a 96-well plate, the heat from your hand actually warms up the pipette, and the volume you dispense will literally drift. It changes. The same thing goes for the surface tension of your samples. Your problem might not be your technique at all. It might just be that you're using the wrong kind of pipette tips for your matrix.
Now, on the biological side, you have classic culprits like the hook effect. This is what happens in really high concentration samples. You have so much analyte that it swamps both your capture and your detection antibodies, which means the assay sandwich can't form properly. And the result? You get this kinetically unstable system that causes wild, unpredictable swings in your replicates, but only at the very top of your curve.
Okay, let's switch gears to a totally different kind of problem. What if your individual replicates look great, but you start to see a pattern, a drift, across the entire plate? Well, that tells you the error isn't random. It's pointing towards something systemic, something in the environment.
And the most common cause of that is the edge effect. You've probably seen this. When you take a plate and move it to an incubator, the outer wells, the edges, always heat up faster than the wells in the middle. That temperature difference creates a thermal gradient, which means the binding kinetics are not happening at the same speed across the plate. And that can systematically screw up your intra-assay percent CV.
Now, here's a source of error that is fascinating because we almost never think about it: the wash step. Every single time your plate washer aspirates a well, it leaves behind a tiny little bit of liquid. We're talking just 2 to 5 microliters. Now, if your washer heads are even the tiniest bit misaligned, that residual volume is going to vary from well to well, and that inconsistently dilutes the next reagent you add, introducing a ton of noise.
Okay, so let's make this super practical. Here is an actionable checklist you can use next time you're at the bench. Ask yourself these questions. Is the failure in a specific row or column? That points to hardware, like a clogged washer head. Is the percent CV really high in your blank wells? That's probably nonspecific binding. Does the CV get better as the signal goes up? We know that one. That's additive noise. And what if your R-squared is fantastic, but your replicates are awful? That tells you the error is probably random and mechanical. See? This is how you start to narrow down the real cause. So once you've diagnosed the type of problem you have, it's time to actually engineer a solution. And that means we have to move beyond just changing one factor at a time. We have to embrace a systematic framework called Design of Experiments, or DOE.
This right here, this is the core mindset shift we need to make. We have to stop randomly trying things and just hoping for the best. With a structured approach, you can methodically and efficiently figure out what's really driving the variance so you can build an assay that's truly robust.
A screening design, something like this Plockett-Berman example, shows you exactly how this works. Instead of running dozens of experiments, you can efficiently test a whole bunch of factors: incubation speed, blocker type, wash volume, even the type of plate sealer you're using, all at once. And the results will give you a chart that points directly to the 1 or 2 factors that have the biggest impact, so you can focus your efforts where they'll actually make a difference.
So, what this all really boils down to is a fundamental shift in how we approach our work. It's about moving away from this idea that assay development is some kind of dark art that depends on one specific person who just has good hands, and moving towards a disciplined, repeatable engineering process.
Let this be your main takeaway. Precision isn't magic. It's not a mystery. It is the direct, predictable outcome of a robustly engineered assay. When you systematically diagnose the noise and isolate the source, you can build a method that's rugged, reliable, and actually works.
So if you are ready to trade in that guesswork for engineering, make sure you subscribe to the Assay Optimization Lab, AOL for short, for more explainers just like this one. We really want to know what puzzles you're trying to solve at the bench, so please drop your questions or ideas for our next topic in the comments below. Thanks for watching.